Hello everyone, my name is Parsin Haji Reza. I'm the Director of PhotoMedicine Labs, and this is my pleasure to be here today on behalf of my team, my grad and undergrad student, my postdoc and staff, to tell you about our pioneering technology, photoacoustic remote sensing, or PARS microscopy. Before I start, I would like to disclose my conflict of interest in illumisonics, which partly supported some of the work that I'm going to show you today. So let's start by defining photoacoustic. In fact, photoacoustic for the first time has been introduced to us by Alexander Graham Bell. He actually used this effect to build a telephone using sunshine. I believe he was not as successful as he was hoping to be because he didn't have access to laser at that time. However, I would say in the last 10 to 15 years, thanks to advancement in the field of optics and photonics and laser, photoacoustic became a hot topic and a very powerful technology in the field of medical imaging. So how we can define photoacoustic? Photoacoustic can be defined based on photon or light as well as acoustic or sound. In a simple word, we can generate sound using light. And then by listening to the sound of light, we can form very unique images. So how that works in the medical imaging field. So in this case, a light source usually is a pulsed laser, a very fast pulsed laser. So this should be either a nanosecond or picosecond or even shorter. The pulse energy in all the cases that I'm going to show you today is going to be very low. It's going to be between nanojoule to picojoule. That means they are very safe. So the idea is this laser need to hit the target. So let's call this laser excitation laser. So our excitation laser need to hit the target. And the idea is the target that we are trying to image need to absorb our excitation laser. So absorption is the key word. So we have this fast pulse laser get absorbed by our target, which means we generate rapid heating, which means basically thermal expansion, and that can be translated to ultrasound signals, or in this case, photoacoustic signals. And then we detect these generated photoacoustic signals using ultrasound gel and transducer in contact with sample in most of the cases. So I mentioned the key point is absorption. So the idea is we need to make sure the wavelength or the color of our excitation laser actually match the optical absorption of our target. For example, in this virtual monitor here, we are looking at capillary beds of a chicken embryo. So in this case, we use a green laser to take advantage of the fact that hemoglobin within red blood cells and eventually within the capillary beds absorbs 532 or green laser stronger than any surrounding tissue. So in this case, only capillary beds start generating sound. And the background is almost signal free. And this is a very important advantage. So what does that mean, basically? That means that anything absorbs light now can be imaged by photoacoustic. And the good news is almost everything absorbs light. And when we talk about absorption, the key point here is label-free imaging. That means we do not need to have any contrast agent or dyes for us to help us to provide the imaging contrast basically externally. Our body naturally provides us with this imaging contrast. So there are a lot of great works out there by my colleagues. For example, it is shown that if we want to take advantage of lipid optical absorption or fat optical absorption, we can use a 1200 nanometer. If we want to basically look at different structure, we just need to select the right wavelength. For example, 1046 has been used to image neurovasculature structure in mouse brain. As I mentioned, visible range, in this case green or 532, has been used to take advantage of hemoglobin optical absorption for us to be able to look at red blood cells or eventually microvasculature structure. We can even go to shorter wavelengths. So for example, we can take advantage of the fact that DNA absorbs the UV wavelength very strongly to image cell nuclei, or even we can start looking at cytoplasm. As I mentioned, everything absorbs light, almost everything. And these are some examples. Even some gas molecules absorb light. So this is exciting because now we have a unique imaging contrast, which is optical absorption. And this is unique because there are not so many other technologies that actually can do that. For example, X-ray, MRI, ultrasound, all those medical imaging techniques that we are familiar with, they are very exciting, very amazing, but they use a very different imaging contrast. So that means using photoacoustic, now we can start doing things, looking at new structures and looking at new information that before actually it was not possible to be able to measure them or image them in the same way that now we can do this. So let's look at some examples, for example. So for example, here we're looking in this image here on the right. This is an image from our group that I'm going to explain a little bit later. But in this image, we took advantage of the DNA optical absorption, cytoplasm optical absorption, and oxy and deoxyhemoglobin optical absorption. And we can actually simply select different chromophores, image them, and unmix them and show them in one image. So now it's very unique because now it provides us with rich amount of information, or a lot of information for several different preclinical and clinical applications. Here, for example, in this video on the right, on the left, we're looking at the oxygenation. This is a functional, basically details that photoacoustic can provide. So photoacoustic can provide oxygenation, blood flow, oxygen consumption, oxygen metabolism, and so many other of those things in a functional way, but very accurately, because now we can, for example, separate oxy from deoxyhemoglobin by taking advantage of the fact that they have a little bit different optical absorption. So we can unmix them. We can start looking at SO2 or distinguish between vein and artery. So in this specific video here, the animal gets stimulated, and based on where the animal gets stimulated, different side of the brain start lighting up. Lighting up means oxygenation. And because we basically can very accurately and quickly measure oxygenation with photoacoustic, we can actually see the brain activity based on the amount of oxygen consumption or oxygenation. And this is very important and very exciting for so many applications in oncology, for functional brain imaging, as well as ophthalmology. So far we talked about all these great features, but as we know, life is not perfect and there are always advantages and disadvantages. But if we could basically think about an ideal photoacoustic microscopy. So please keep in mind, in this talk, our focus is on optical resolution photoacoustic microscopy, or basically a microscopy version of photoacoustic imaging techniques, because we really want to look at the for example, single cell or cell nuclei, because we want to focus on high definition imaging capabilities. So that ideal photoacoustic system, in my opinion, should be non-contact, like any other microscopy techniques we know. For so many reasons, for so many applications, we do not want to touch the tissue. Let's say when we imaging someone's eye, or when the brain open, during the surgery, when you have basically some targets in your preclinical studies for many valid reasons. You do not want to touch them, you do not want to deform them, you do not want to disturb them. However, unfortunately, as we discussed in photoacoustic, we always need to have that ultrasound gel and transducer in contact to be able to detect the generated photoacoustic signals. We want to be as small as possible when it comes to footprint. Ideally, we want to make macroendoscopy systems. So we want to take this very high resolution imaging modality inside of body cavities and be able to image. So that bulky ultrasound transducer doesn't really help. In microscopy, usually the penetration depth is limited because the focus is on high resolution imaging. So it's always ideal if we can actually keep that high resolution capabilities, but push the boundaries of our penetration depth. So to give you a ballpark number here, usually using green or visible wavelength in scattering tissue, for example, the skin, we really cannot focus more than 1 millimeter into the tissue. That means our penetration depth usually is around 1 millimeter, again, in scattering tissue. For example, for eye, because it's clear, we can penetrate and image the entire eye. But in scattering tissue is around 1 millimeter. So the ideal scenario would be if we can increase that penetration depth even twice or 3 times and still have the same resolution. We want to make sure that we can image any target with any thickness. So for example, during the in situ imaging, when you image someone's, let's say, body, you may want to penetrate a little bit deep, but you don't want to care about how deep the target is or the sample is, because if that's the case, and you only be able to image teeny tiny samples, then translating that technology to clinical setting is almost impossible, because if you want to image someone's eye or brain, or do some, let's say, in situ imaging of someone's basically body, that is almost impossible. And if we have an all optical imaging system, in other words, if we didn't have that ultrasound transducer, then our microscope, this very exciting microscope that we show how important it is, how novel it is, and how unique it can be, can now look like any other microscopy systems that we are familiar with, which usually are non-contact. You do not see any transducer. You do not need to touch the sample. Usually there are some lenses and the sample, and basically that's it. If we have that, now we can easily integrate our technology with other optical imaging techniques out there. For example, optical coherence tomography, fluorescence microscopy, Raman microscopy, and other modalities out there, which can add a lot of benefit, provide us with basically complementary imaging contrast. So before I start now introducing PARS technology and explain how PARS can be an ideal photoacoustic, optical resolution photoacoustic microscopy, let's just remind ourselves how general photoacoustic works again. So the excitation laser need to be focused. As I mentioned, usually it cannot penetrate more than 1 millimeter. And the laser going to get absorbed by the target. So in this case, let's say we have a red blood cell. The absorption going to cause some sound or ultrasound. And then you are waiting for ultrasound signals come to you at the surface using ultrasound gel and transducer. So basically what we measure is ultrasound pressure propagation. So before I start how PARS works, and the idea of this talk is to make it as less technical as possible. So I'm going to give you an analogy that I always give to my students. It's not a perfect example in terms of physics, but it really will help us to understand what is PARS really. So for some strange reason, some of us, when we go to camping or when we go to nature, if there's a lake or pond, we throw a stone inside into that lake or pond. So if you think about it, in that exact moment that the stone hit the surface of the water, we do have this initial pressure, this shock waves that eventually generate waves coming to the shore. The further we are from that impact point, the less pressure we are going to detect. The closer we are, the more pressure we're going to detect. So now imagine if you are at that exact moment that stone hit the surface of the water and detect that initial pressure, that shock waves, then you are dealing with orders of magnitude higher pressure compared to the time that you're waiting at some distance for the sounds just basically decay and propagate, and then you detect much, much, much less amount of pressure. So if we basically replace that stone with our excitation laser, the waves we are basically generating is of sound, or it's of photoacoustic signals. And there are two ways for us. One is waiting at the shore with ultrasound transducer gel for the sounds come to us, or use PARS technology and go to the origin, only collect few hundred nanoseconds, or just collect the initial pressure at every time that the stone, in this case our pulse laser, hit the surface of water. So in other words, with PARS, we want to detect only initial pressure, those shock waves. Of course, we can be sensitive to propagation, so many other physics. In this talk, I'm going to make it as simple as possible. So the idea is now we want to replace this ultrasound transducer with another laser. Now we call this a detection laser. And this laser is co-focused and co-scanned with our excitation laser, in which we only want to detect the initial pressure. So this is a new physics. This is a new way for us to form images. So we take advantage of optical absorption and a little bit changes in temperature and pressure to look at the changes in optical properties of the material in which our detection laser is going to be affected or modulated based on basically the initial pressure and the initial temperature that we basically generate at the origin. So this is very exciting. This means now we have a non-contact technique. We can have few centimeters of working space. Again, PARS has very different modalities. We can actually detect initial pressure as well as the surface oscillation due to traveling of this ultrasound pressure propagation. But in this case, we only focus on one of the simplest methods. So how that has been invented? Basically by accident. When I was doing my PhD at the University of Alberta under Dr. Roger Zem's lab, basically, I think it was third year of my PhD, I was working on a very different project, and all these years still, I always like to test different things that usually never works and doesn't make sense. So accidentally, during one of those tests, when I was bored with the experiment, I realized when the ultrasound transducer that I was designing that time, when it was not even on the sample, I still could see some level of signal. It was very weak, really looked like noise, but meant something at that moment to me. And then, of course, it was so weak that for the first few weeks, no one believed me, my colleagues, my supervisor. They really thought I'm crazy. That doesn't make sense, because we couldn't really explain how we can detect sound through air, because we didn't really considering this effect. And it took me basically a few weeks to be able to convince everyone that actually this makes sense, and I could actually get some images. Then I spent the rest of my PhD, my entire postdoctoral fellowship, focusing on this technology and take it from a very weak concept to something that I'm going to show you today, of course, with help of my colleagues and all the people involved. And then since in 2018, I joined University of Waterloo as a faculty member, we built an amazing team. We came with very, very decent and detailed plan. And we really took this technology to a very different level now that I'm proudly going to show you some of our latest results. So I mentioned that using photoacoustic, we can do multiplex imaging. We can select different chromophores by taking advantage of the optical absorption and show them in one image. So one of the very important things we do in photomedicine labs is actually building a very strong preclinical lab. So we build, for example, chicken embryo model in-house. We're designing our own cell culture models and a lot of different preclinical basically experiment in-house. So this image actually taken by PARS by taking advantage of, as I mentioned, 4 different wavelengths on the chicken embryo. So we can look at DNA, cytoplasm, oxy and deoxyhemoglobin optical absorption to form this level of images. So this work has done by postdoc Kevin Bell in the lab. He also took this to a very different level and tried to, again, image this chicken embryo model with 4 different wavelengths again, 250, 420, 532, and 560. So the 250 nanometer here show us actually the cell nuclei, and 420 to 530 to 560 actually showing us oxy and deoxyhemoglobin, basically optical absorption. We haven't unmixed them to show you SO2, but in the next slide, I'm going to show you some of this SO2 or oxygen consumption, oxygen saturation measurement. But these are very, very important because this is a live animal, and this has never been done to the best of my knowledge, that on a live animal, we can actually do this level of multiplex imaging. And now you can imagine the amount of research this system can open in pharmaceutical development, in biology, in preclinical applications. So these are some of the oldest images that I took when I was a postdoc at the University of Alberta. But I think they are still important for me to show you here. So here in this image, we're looking at the structural information. So this is microvasculature of mouse. We can look at capillary beds, bigger blood vessels. We can zoom in a smaller area. We can see smaller targets, or we can zoom in one capillary and see red blood cells. We can see these red blood cells actually in real time as well. We also can do functional imaging very accurately, and I'm going to mention why very soon, why photoacoustic and now PARS is actually much more accurate, to best of my knowledge, in my opinion, than any other imaging modality out there. So the idea is, for example, here, we image the same field of view, at least with 2 different wavelengths, and take advantage of the fact that oxy and deoxyhemoglobin have different optical absorption. So we can actually distinguish vein from artery, or figure out which part is more oxygenated, which part is less oxygenated. Huge for oncology, for ophthalmology application, which I'm not going to really go through a lot in this talk, but I'll let you to think about it. And I'm sure many of you are familiar with some of the applications. But the idea is the more reddish here means more oxygenated, yellow means less oxygenated. So more microvascular structure of the mouse ear. And this, in this case, again, we use a green laser to take advantage of the fact that hemoglobin absorbed is stronger. And this is why the background is almost pitch black, because this is an advancement or advantages of photoacoustic, because we only make sure the targets that we are hoping to image generate pressure or sound, and we only want them to shine for us to be able to detect them. This is the vasculature of the top of the mouse ear, or tip of the mouse ear, and you can see how nicely we can actually see that. So in all these cases, animals are alive. They just basically go under anesthesia, they sleep for a few minutes, we image them, they wake up and they leave the lab. So we can simply actually do this with very low power and a lot of care about our animals. So one of the very important things I wanted to do when I started my career in Waterloo was to actually design a accurate real-time feedback or real-time display because that would help us to understand the physics, the mechanism better, help us to do better alignment. And in this case, we're looking at the mouse ear on top and carbon fiber networks on the bottom. So carbon fiber networks basically consist of several single carbon fibers. Each of these carbon fibers are around 6 to 7 micrometer diameter. So what you're seeing here, we're moving the target in X, Y, or Z direction, and we can easily in real time track them, which is very helpful when we really take this to eye imaging or more complex structure. So this is done by my master's student, Sada Bassi, which he did an amazing job when he was working in our lab. Then we show for the first time actually the 3D capability of PARS system. And these are actually, again, carbon fiber networks. 6-7 micrometer is the diameter of each of these carbon fibers. And we can actually now image them in 3D. Again, this work done by Sada Bassi in our lab. We can do this 3D imaging in animal or microvasculature as well. So what PARS now can offer? We have a very unique imaging contrast. We can now image optical absorption as well as optical scattering. Why optical scattering? Because our detection light by itself can actually perfectly act as a confocal scattering microscope. And when the excitation is on, now we can directly measure optical absorption. So it's a luxury, it's an advantage for us that general OR-PAM or optical resolution photoacoustic systems, they don't have it. We can have a very high resolution. Resolution usually is wavelength dependent, but we can achieve diffraction limited resolution in most of our cases. So the lowest resolution we could achieve, for example, was around 250 to 300 nanometer, which is more than enough for us to be able to look at cell nuclei. But we are working on even super resolution methods, and we do have some patterns on this idea. Because of the fact that now we have this very unique detection laser, and the detection laser in many cases, in some cases, we actually will care about the fact that our detection laser also get absorbed by the target, but let's ignore that fact and imagine if we want to increase the penetration depth, we can actually select our detection wavelength to be actually long penetrating wavelength. So in other words, the longer the wavelengths, usually they can penetrate deeper. And in this case, our resolution can come with, from the focused detection laser instead of focused excitation laser. And we show in our optical paper that I cited a few slides before, we can actually do 2.5 to 3 millimeter basically penetration depth. And this is still in phantom, but compared to, let's say, 0.5 millimeter to 1 millimeter, we believe that by just selecting the right detection wavelength, only that, I mean, there are other things we can do and we are doing, we can increase the penetration depth a few times, which is very important. We can do real time, as you saw, and because we not worry about the basically time of flight of ultrasound or sounds come to us, because the speed of sound is much smaller or much actually slower than the speed of light. Usually the speed of general photoacoustic systems need to be limited based on the sound propagation. In this case, because we do not really deal with sound propagation, we can really improve our imaging speed by several orders of magnitude. Again, I cannot really go deep in all of this. We can do very accurate functional imaging capabilities that I'm going to show you in the next slide what I mean. It's very important. Now, because we do not have that bulky ultrasound transducer, we can actually reduce our footprint to be as small as a single mode fiber. And this works in reflection mode. That means doesn't matter how thick the tissue is, as long as you penetrate, let's say millimeter, 2 millimeter, 3 millimeter inside that tissue, you can image it. So that, again, reflection mode means you excite from the top, you detect from the same surface, so it doesn't matter how thick the tissue is. In some optical imaging modality, if you excite from one side, you need to detect from the other side, and that means the sample needs to be very thin. So this is not the case for us, which is very important. So let's quickly look at this figure here to explain why PARS can actually provide us with better, basically functional capabilities. So the first idea is there are technologies, for example, optical coherence tomography, OCT, that also try to measure optical absorption, but they do that indirectly. So what does it mean? They have this laser, or the beam they send to the sample, and they collect the reflection of that laser. So the less light reflecting back or scattering back, they assume more absorption happening in the sample. And this cannot be very accurate because the size, the shape of sample can really affect the amount of scattered light coming back. Also, in subsurfaces, there might be different structure as well to change the amount of scattering. So it's not a direct measurement of optical absorption for us to do functional imaging, to do SO2 oxygenation, all those things. In photoacoustic, we have a luxury, or let me correct myself, in PARS microscopy, we have a luxury of having both type of measurement. First of all, if we forget our absorption or excitation laser, our detection laser can act the same way as OCT. However, now we have an extra luxury, and that is our excitation beam, that we can select it very carefully to look at the differences between, let's say, oxy and deoxyhemoglobin. We can send that to really probe very specific molecules. And in this case, now we really more accurately can image or can measure SO2 oxygenation, absorption. Maybe the simple word is to measure absorption, and then absorption can be transferred to oxygenation and functional details. But and this is the exciting part, because now we are sensitive by two different mechanisms. One is direct absorption of our excitation beam, and the other is basically an indirect measurement of our detection beam. So to just give you an overview of what I explained today is very, very shallow. We have several different architectures. We have non-interferometric PARS, we have interferometric PARS. We can use camera to detect PARS signals instead of photodiode. We have coherence-gated photoacoustic remote sensing, which give us depth information. We have single source technology, SD, SE. I'm going to skip them. We have a lot of actually patented architecture, a lot of designs that every day in the lab we actually developing them. So this is a very brief overview of what PARS can do. And as I mentioned, there are 7 family of patents are already filed or issued actually internationally. So far what I show you was new capabilities of a novel optical imaging modality. And as we can all imagine, now we can apply that to several different applications, from preclinical application to pathology or histology, which I'm going to explain soon. We can use this for surgical guidance, for eye imaging, ophthalmic applications. Now, because we do not have this bulky transducer, the footprint even is going to be very tiny. We can use it for endoscopic application. And here I only show a few examples. Basically, this can be extended to, I mean, in my office, I actually have a list of 40 to 50 applications that actually with PARS we can focus on. But for the purpose of this talk and for the simplicity, we're going to focus on one or two applications. So let's talk about histopathology or pathology first. So it's important for us to understand the workflow before we jump into this a little bit deeper. The idea is if there is a tumor that must be removed, the doctor has to make sure they remove the entire cancerous tissue down to even the last cell, because if you remove any cancerous cells behind, the tumor can come back. And unfortunately, in many cases, there is no way during the surgery very accurately we can measure the tumor margin. And that's why, let's say the study shows up to 10% of all solid cancer, they need to go to the second surgery. For example, in breast, there are some studies shows that up to 60% of the cases, the patient need to go to the secondary surgery because in the first try, we don't know how much to remove. So there are intraoperative ways, for example, a frozen sectioning, but here I'm looking at the gold standard. So the gold standard, which is basically a H&E paraffin embedded basically method, is the fact that eventually we need to remove the tissue. Either this biopsy is during the surgery, is open surgery. Eventually you remove the tissue and you end up with resected tissue, is a fresh resected tissue. The gold standard, again, is to put this tissue inside paraffin. And the paraffin molecules will replace water molecule of the tissue. So you end up with a reserved tissue within paraffin wax. So the tissue can last much, much longer. So the idea is we need to shave the surface of this tissue block and put it on glass slide. And if you put this glass slide under general microscope, you cannot see anything because as you can see, the tissue is colorless. So we need to add colors. In this case, two very well-known stains, which hemotoxin and eosin, or H&E, are basically the gold standard in many, many cases. So hemotoxin target cell nuclei and eosin targets cytoplasm. And then pathologists put this under a microscope, and then based on the size, the feature, the shape, the structure of all this information, they call the doctor back and they tell the doctor that, yes, it's tumor, it was cancerous, and secondly, did you remove the entire tumor or not? The idea is they're looking for the rim of the healthy tissue around the cancerous area. And if you do not see that, that means the cancer left behind. So this work typically takes around a week, sometimes even 2 weeks. Again, it can be shorter, depends on the hospitals, but it is at least a few weeks. So the gold standard is here. This is where we are right now, everywhere in every hospital. Our goal is actually go ahead and give the same exact H&E images. We are not talking about the signal. We are not talking about the different measurement. We are talking about the same exact image that all these pathologists and all this department and all this basically histopathology field, expert in the histopathology field, has been trained and comfortable to look and analyze and understand. Ideally, we want to do that in situ during the surgery. Obviously, this is a very big goal and very challenging goal. In order to achieve that, we have some objectives. The first objective for us is to make sure that we are able to image each of these intermediate steps and get the same H&E-like images that later from basically H&E glasses slide the pathologist can get. We want to make sure our laser and our system is very safe, that we never interfere, we never damage any of this workflow. So the workflow can move on, so our technology can be added on to any of these intermediate steps. we want to make sure we have a very good resolution that match perfectly with the gold standard. The ideal case is we want to image the entire tissue in the fastest possible way, even less than 10 minutes, even less than a few minutes, rather than waiting weeks, hopefully to do that in few minutes, and provide the same, as I mentioned, exact H&E images. And we can actually go beyond H&E because now we can maybe add a lipid contrast or different proteins contrast, because as I mentioned, H target cell nuclei, E targets cytoplasm, but what about if we add fat and maybe even hemoglobin or other targets and be able to go beyond H&E, which is still very exciting. So I'm going to show you some of these images. So this is the work done by Benjamin Ecclestone, a PhD student in the lab. He's really doing amazing. He takes this technology to a very different level. I'm going to show you a lot of results in the next few slides from Ben. So this is one of the very first images. So we started by taking one step at a time. So we said that we have this glass slide, standard H&E glass slide. And we thought, can we image unstained slide using PARS and get similar information for us to practice and to understand this technology better? So this is a human brain. And this is the first time anyone used this technology on human samples. For example, Rogers and groups in Alberta that, as I mentioned, I invented this technology when I was working. After I left, they worked on some tissue samples, animal tissue samples, but we really built this technology to be directly applicable to humans and really reach to that level of clinical, actually, quality, so we can move on with clinical, basically, studies. So this is actually a very, very bad case, because this is, here we actually seeing this positive surgical margin. This is not the perfect way that is defined in brain tumors, but for the simplicity, let's call it positive surgical margin. In other words, we see the cancerous area at the rim of this removed tissue. That means some of this cancerous tissue left behind. So keep in mind, these are adjacent slides because the first slide has been stained, and then we shave another slice, didn't stain it, and then image it with PARS here. So with both cases, we can actually distinguish the same structure. So in order to get H, we use a wavelength, mostly a 266 or 250 nanometer, by taking advantage of DNA optical absorption. And in order to get E, we use around 420 to 450 nanometer to take advantage of the fact that cytoplasm absorbs stronger in those wavelengths. So it's the same exact way to target them, because H&E labeled them in unstained tissue. We actually select the right color of the wavelength to image them at the first place. So we really can get very accurate measurement of H&E or cytoplasm and DNA. In this specific image, we only use one wavelength to look at the cell nuclei structures. However, for example, in this specific case, which is a still human brain, still adjacent slides, we use two wavelengths for PARS to get both H and E. And as you can see, this is the gold standard here, and this is PARS. Again, the structures are not perfectly matched because they're adjacent slide. When you shave it, you go to different cellular morphology and cellular level, but you can look at all those information that highlighted by clinicians, which matters when it comes to diagnosis. So it's not still one-to-one, which I'm going to show you very soon. More of this H&E and PARS one-to-one comparison, again on brain. And if you let them actually overlay, even though they're adjacent slide, we can see how nicely PARS images now can actually resemble H&E gold standard methods. Before I move on, I'd like to mention again that we can now build an endoscopy method to do virtual biopsy, to take it to body cavities, and actually in real time, hopefully, get H&E images. So we did also work here to look at the gastroenterological samples and be able to test our system to see how it works on the GI or gastroenterological sample. This is another work. This is done by Kevin Bell, the postdoc in the lab as well. He's the one actually for the first time showed H&E images by you taking advantage of 2 different wavelengths. So it was very exciting. So these are all PARS images. Here we're imaging the entire human skin. Again, still on stained tissue slide. If you put this slide under normal microscope, you do not see anything. And by taking advantage of the absorption of different basic chromophores here, we can form these images. We zoom in a smaller area. Then we use 2 wavelengths. Here we only use one.
You'd like to call in.
Sorry about that. So here we only use 1 wavelength, which is the DNA wavelength, but then we use 2 different wavelengths, DNA and cytoplasm, and we get H&E-like images. And then we focus on a smaller area and actually be able to see the human cells. This is from Mo's sample. Mo's is a very precise way to look at skin cancer. I'm going to skip the explanation of Mo's, but I encourage you to look at it. I'm sure many of you are familiar with Mo's samples. But the idea is now use PARS, only 1 wavelength, to get the entire sample. And the idea is to do it as fast as possible. So for some of these images, now we can do it around 10 to 20 minutes, depending on the resolution. But with some advancement, we can take this to less than 10 minutes or even down to a few minutes. So again, these are all skin cancer images. So we decided to make sure that we test our technology to be able to validate that it's the level of pulse energy that we use is safe. We use in the range of, let's say, 600 to 800 picojoule, but we still wanted to make sure this is safe. Of course, we can even reduce that with more advancement. We are working on it. So what we did, we grabbed one of these unstained glass slides. First, we imaged that with PARS with 1 wavelength, and then we stain the same exact slide. So we get the first ever 1-to-1 true comparison. And then we see how nicely, actually, if we zoom in different area, this structure now can actually match and be able to get this 1-to-1 comparison. In other words, also by looking at H&E images after PARS experiment, we can see the cells and we can actually make sure the system was safe and didn't damage the cells. And even if you overlay them on top of each other, we see that how nicely they can actually match. So this is very important for 2 reasons. We got 1-to-1 comparison, and then we showed that PARS images are actually safe to be used. I'm going to be very quick here, but so far to get H&E images, we use 2 different excitation lasers. However, as I mentioned, we have a scattering imaging contrast as well. And we took advantage of that fact as well, because now we can make sure our excitation target DNA and let's say our detection target basically cytoplasm. So by looking at basically reflection or scattering of our detection, we get E. By looking at the absorption of our excitation, we get H, and then we get H&E-like images with 1 excitation wavelength. That means much, much faster. So this is H&E. This is PARS H&E images based on both absorption and scattering, which is another important thing. So in this slide, very quickly, I want to make sure we understand the fact that we can do the entire field of view very quickly with PARS. We can zoom in a smaller area and see this level of cellular structure in a very dense, basically cellular morphology area. And this shows the quality of the resolution and signal to noise ratio that we can image. So this is still human Mohs samples and important for us to be able to see. We have done very, very exciting and very true clinical studies on most sample. The result will be published hopefully very soon. But we work with a lot of pathologists and dermatologists to be able to actually study or pass images from moles. So there are some results here that we are going to do. So these are all human samples, and now we can actually look at all the cellular structure. Again, this is some clinical studies that I'm quickly going through. So for us to be able to see the level of the study and the quality of images that we can achieve using this technology. So the whole slide, which is in the middle, this is a breast cancer actually, or a breast sample actually, a human breast sample. We can again do that in hopefully less than 10 minutes or even less than a few minutes. And then we can zoom in a smaller area. For example, red is here, orange is here, blue is here, and green is here, and see this level of morphology. To make this a little bit easier for us to understand, Ben Beckelston actually done all this work, recorded a video for us to be able to see. So this is a human breast. And we zoom in different area into almost half of this glass slide to see the level of the structure and resolution we can achieve. And if we can image the entire tissue with this level of resolution, less than a few minutes, I think that is ready very quickly to be adopted to a lot of different applications. So this is very exciting for us because this is never been done and very unique. And now we can even expand this to beyond glass slide. So far, we only show glass slide images. So as I mentioned, we can get black and white in PARS images. We can get PARS H&E-like images by taking advantage of both absorption and scattering, or by taking advantage of two absorptions, or even more excitation beams to go beyond H&E. I'm showing a different images of human breast again to show that we can actually do both black and white and color version. So the next biggest step for us to now move this for the first time and image thick, unstained, standard, paraffin-embedded human tissue. And these are breast tissues. So what we've done, we actually shaved the surface of this human breast, send it to the gold standard pathology H&E. We got the surface of this basically on glass slide, and we have H&E of it. And then this is very important. We directly image for the first time the tissue block. And then we wanted to get match images from the tissue block. This is very important because each of these hospitals and pathology departments, they have hundreds or thousands of these tissue blocks. So if we can directly image them, we hopefully can save a few days. And also this speaks of the quality of the system in terms of signal to noise ratio and resolution, because this is very difficult to image. So this probably give a better idea. The top is H&E standard images, again, by shaving the surface. And then on the bottom, we directly image the tissue block. Of course, they are not perfect match because we directly image the tissue block. They are adjacent. However, we see how nicely we can see the important structure that matters. And again, to the best of my knowledge, there's no technology in label-free can image the non-contact label-free this level of quality and get these images. So I mentioned we can do H&E images using one single excitation, and by taking advantage of the fact that then the detection also absorbs light, and detection also gets some level of contrast for us and provide us with some scattering. And this is a standard H&E from the glass slide. And again, we image the tissue block directly. The next slide, or basically the tissue block directly, and the first slide shaved went to the H&E to get H&E-like images for the first time from glass, from the tissue block, from the surface of tissue block. And to me, this is very exciting because we're really coming to this quality that we may actually now skip the glass slide and directly image unstained paraffin embedded tissue block. And anyone works in optics, they can relate to the fact how difficult it is to focus and collect information within paraffin, because paraffin is also absorbing light and they are super scattering. So if we can do that, and we can actually get H&E-like images, again, by taking advantage of both the scattering and scattering and absorption, this really matters. I'm quickly going through, again, black and white PARS, H&E-like PARS. So the good solution that also we can offer, or exciting solution that we can offer, we can virtually actually slice this tissue block instead of using basically a mechanical shaving as the gold standard actually usually take advantage of, and put each of those shave slides on glass slide and then do H&E on them. We can image the tissue block itself, and then by changing the focus, do virtual slicing. And here we're going through depth. We actually flying through depth and see different cellular morphology in paraffin embedded tissue block, which is very exciting. So Ben Ecclestone again, he also provided us with some videos here, so we can appreciate the fact that we can actually get the same level of information now in tissue block with this level of resolution. Of course, a lot of image processing, a lot of exciting things can be done, but this has been done almost in a year, in the last years, knowing the fact that COVID has really hit the lab and all experimental works. We really came a good path, and a very strong result has been basically produced thanks to amazing things that we have. Then we moved to resected tissue. I wanted to directly image the resected tissue. So this is the entire kidney of a mouse. We image that by PARS, and then we zoom in different areas. And now by taking advantage of two different excitation wavelengths, one to target DNA optical absorption to look at cell nuclei, and other one to image cytoplasm, we can for the first time actually get H&E-like images from fresh resected tissue. Another important thing we have done, we built the OCT optical coherence tomography system in a very unique novel method that I'm not going to disclose right now into our PARS system. And the idea is with that OCT method, we would like to get a 3D morphology or topographic of our surface, because as you can imagine, as we can see, we do not have a flat surface. So it's important for us that we can adjust our basically focusing capabilities either by using adaptive optics or mechanical focusing or some optical tricks to be able to look at different area in resected tissue. And this is what we've done. So this is actually tumors in a rat on a resectedly fresh tissue. And for the first time, we can actually see very relevant information that our pathologists and our clinicians approve that these are exact information they were hoping to see from actually a tumor in a resected breast rat tissue. As I mentioned, this OCT work is to be able to look at different morphology of the tissue is in progress. We're really pushing this to a very different level. So this is another example, again, of imaging this rat tumor in the breast, and then we can see the entire field of view of this, again, resected tissue. Sometimes this resected tissue needs to be dipped inside some solution to make sure it's not expired so quickly, and sometimes even directly after the measurement, let's say 10 to 20 minutes without even touching that, we put it under PARS microscope to get these images. So we can zoom in a smaller area and we see we can reach to this level of resolution, which in this case around 200 to 300 nanometer resolution, to see all this different cell nuclei structure, which is very important. Another exciting things we've done, and this is very, very new work done by, again, Ben Ecclestone, we grab one of these resected tissue, and then, as I mentioned, we virtually slice them, and we go to different slides, and we can actually, for the first time, get 3D level of images from fresh tissue. And all these things really can add up for the future, hopefully, goal or ultimate goal we have to either have a device beside the surgeon. So immediately after the surgery, they put this resected tissue, get all this information they need to finalize the cut, making sure everything's removed, or even one step even above that, to have a surgical microscope that in situ, we can actually directly see and cut and remove the entire area. As I mentioned, we can go beyond H&E. So here we start looking at actual lipid contrast in the pig tissue and chicken tissue. And these are also very first time. PARS has been used to actually look at this level of morphology on basically tissues. And this is very, very exciting. I'm also very excited for the first time to show that we have worked on freshly resected human tissue. And this is, again, done by Ben Ecclestone. This is the human breast, just basically resected. It's dipped into the solution because we don't want to make in the formalin fixed solution. We don't want the tissue to get expired. But we put the entire thing like this under the microscope, and for the first time, we get histology-like images from resected human tissue. Of course, these are very new. We still need to push it and improve it, and a lot of clinical studies need to be done. It's still at the very early stage, but very exciting stage, because in a few years, or maybe less, depends how fast we can move on, we may have some solutions to move on. Another thing I want to quickly cover, we are almost at 1 hour here. So the idea is we want to make sure that we could image we want to make sure we can combine PARS, as I mentioned, with OCT. And here at the B, we're looking at OCT images of a chicken embryo. We can look at different blood vessels. And when you cut through, you can actually see the cross section. E also is a PARS image, but OCT image, but C and F are actually PARS images from this chicken embryo. So why we combine PARS and OCT and we start imaging chicken embryo? Because OCT is the gold standard in ophthalmology. If I wanted to move to ophthalmology, we needed to work with OCT to understand our technology better. So we took this technology and also imaged mouse ear. And in this case, A and B actually show OCT images of the mouse ear. We actually cut through, thanks to the resolution we have. We can see different layers of basically skins, and then we image the same area with PARS. To see microvasculature structure, we zoom in a smaller area, and we can see single red blood cells, which is very exciting. So I need to be quick on this, but a lot of these blinding diseases, including glaucoma, DR, AMD, they basically do not have any early signs. And sometimes when we diagnose them, it's a little bit late. If we can do this at earlier possible stage, so we may be able to change the way we practice them, the way we manage them, and the way we treat them. There are studies show that the amount of oxygenation, SO2, and even metabolic range change of oxygenation, oxygen metabolism, really matter when it comes to any of these blinding diseases. The issue is there is no technique right now in the clinic that can measure those amount of functional details in eye for us to be able to move forward. So you saw that we can do a very good job to image some of those things with photoacoustic, in particular with PARS. And now because PARS is the first non-contact photoacoustic techniques that showed all these capabilities, now we can actually take PARS to the field of ophthalmology. We just so new in this field. Actually, for the first time, we published the very first ever report on this work, which is, I hope, this the starting point of a very big era for the technology and hopefully for the field of ophthalmology. But as I mentioned, we combine that with OCT. We image this is an animal eye. This is a mouse eye with OCT. So we can see all these important layers. And then for the first time, we use PARS to look at these images. So we can see basically in CD, basically the iris structure, the blood vessels on the iris. In E, we can actually see back of the eye. This is a retina. And then we try to image them with several different wavelengths, in particular, the iris blood vessels, to show for the very first time, actually, non-contact SO2 measurement using optical absorption contrast. And this is done by Zohar Hosseini, Nima Abbasi, and Alex Siemens and a lot of other guys also helped with some of this work. So very exciting. Is a very early stage, but very promising. This probably need to very soon, hopefully with some quick tests, be moved to bigger animals and hopefully human studies. I just want to quickly show two images here before I wrap up this talk. The combination with OCT really help us to understand where we are, what we are doing in the eye. For example, we build it in a sense, as I mentioned, this is a unique way we've done it. I didn't really have time to disclose it, but we can see the entire eye. For example, we can see the top of the eye and retina, so we can be able to put our system or our PARS system in a place that we really be able to understand. This is another cool image done by Zohar Hosseini. I just wanted to show you here. This is actually the eyeball of the rat from the top. It doesn't really mean anything from the clinical point of view, but I thought it's cool. I just wanted to show you as the last image here. I like to thank all our funding sources to supporting this work. This is an exciting journey for us. Everyone in the lab, one of the best possible groups that I could hope for and I could dream for and I could imagine for. I'm very thankful to each of these guys for being such amazing, hardworking and intelligent and passionate people. They all share one passion, and that is to make a difference and make an impact in the field that we are and hopefully into our society. So I'm very proud to be part of this team. This is an old image. It's the pre-COVID, before COVID. Hopefully we have after COVID images, but we have some, I try to gather some of these images together. Everyone working in our lab in last year. I do have the entire list of anyone working in the lab. So we started our lab officially from January 2019. So it's not very old, but the result and the contribution of these guys really took our lab to a very different level. I'm thankful for all of these guys, undergrad, graduate student, postdocs, RAs, staff to make contributions to this amazing work. All our collaborators, all clinicians and my colleagues at University of Waterloo, thank you for being who you are, and thank you for all this impactful contribution, amazing conversation we have. I would like to mention that we're always looking for good people, talented students, researchers, anyone that wants to be part of this journey and they are actually ready to make contribution. Please feel free to send your CV to me. There are a lot of open positions all the time in the lab because we're growing very fast. With that, I would like to thank you everyone for your attention. This is E7, Engineering 7. This is where all our labs and offices are located at the University of Waterloo. I'm very thankful for this support. If you want to know more about what we are doing in our lab, please feel free to look at our website. Also, every year we have a progress report for public. So feel free to check out our 2020 progress report in December. We will have for 2021, and every December we try to keep this habit to have public report for anyone who's following and interested in our work. So with that, I would like to thank everyone, and I'm more than happy to answer any questions. Thank you.